stimulus will causes the secretions in the oral cavity through this particular exocrine part of the gland. Now, hormonal, for example, the secretions from the hormonal, as it is the gastric influences when you eat something that stimulus and which leads to for the purpose of the digestion, as the hormones will secrete within the abdomen. That is one more best example. Now remember the myoepithelial cells, myo pertaining to the muscle. Epithelial, it means wherever there is an epithelial cells are there along with these myoepithelial. Beneath the epithelium there is a muscle filaments or muscle smooth muscle cells will present, and they help to, for example, for what they are, the shape of their star shape, they are contractile in nature. It means the whenever the myoepithelial contract, the secretions within the cells which get bursted and comes outside. So that is the best example we will discuss. I will show the in the figure how the myoepithelial cell. But just you remember these myoepithelial, the shape is star shaped. They are contractile in nature. They lie between the secretory cells and the basement membrane. They are not in the apical region. They are there in the basement membrane. The reason is whenever the contraction over the basement membrane takes place, automatically the apical part will get bursted and secretions within the cell releases within the tubular area that I will show you in the figure. So this is how the myoepithelial shows. So these myoepithelial cells we used to call serous deminules. Remember, this is very important. So when we discuss in the histology practical class, at the time we will show you this how the serous deminules will present. So the myoepithelial cells or the basement membrane of the your cells. So this is serous deminules, right? The cell or the cell is used to call serous deminules. Now, before we start the particular exocrine gland, I would like to tell you the development of this particular, as these glands, usually the duct glands are the exocrine glands which develops in our body as their invagination of the epithelium into the underlying vascular connective tissue. Remember, so that particular glands are ducts, they are invaginations within the epithelium and underlying vascular connective tissue. The vascular connective tissue is to nourish the deeper aspect of, and it not only nourish it, whenever there is an effector stimulus will taken place, hormonal action, that part can be taken till. So during the development these vessels are reaching to the peripheral part or the end part of the epithelial layer, whereas the distal part when you take the, it forms the glandular or secretory end piece, functionally an active portion. The meaning is that the lower segment or inner segment or most peripheral segment part of the glandular area are secretory end piece of this particular, so they are functionally an active portion, means the whole gland and duct as well, it is an active part which secretes continuously. Your glands means tube means it may not have just a tube, it is tube contains again cellular element that also secretes to prevent the damage of the whatever the secretion is coming, or else the secretion along with it slowly move from one end to another end, which helps. That is the reason. So this is a from ascending to till the distal part, it is an secretory, whereas the proximal part and excretory duct opens into the surface of the epithelium, and some cells get detached from the epithelium surface, that is we used to call ductless or endocrine glands. It means the single glands will be there in between the here and there, wherever we used to call, for example, if you take the pancreas, we have isolate of Langerhans. So those glands, the secretion directly reaches through the their effective organ through the blood vessels. So that is one of the endocrine gland, but which is completely constituted present within the exocrine gland, that is your uh, pancreas. Now the development, when the development takes place, what happens? So there is an bud. So bud, so there is an, this is the epithelium. So the bud formation takes place. These blood migrate the cell towards the one end, and this particular migration of cells along with the clusters area, this particular clusters area, so which forms the multiple cellular elements. So those cellular elements divides into the upper portion is a ductule, and the invaginated part which we used to call the the glandular area. Now the glandular area divides several cells in a different, either it is in the form of columnar or in cuboidal or in the form of apical, that is we used to call pyramidal cell shape, different shape will, and nearby this particular there is a homogenic area will be there, so that particular homogenic cells will develop slowly nearby this particular, we the capillary network will develop over here. So that capillary network will fuses through the basement membrane of this particular cells, and which completely constitute form a complete the, this is an endocrine gland, and rest of other is the exocrine. This is how the development of this particular gland takes place, right? Now the glandular epithelium, when we say, so this is the epithelium in the form of column cuboidal, or else this is a columnar, say this is the glandular columnar epithelium. So sorry, this is the when centrally placed the nucleus, and the unit completely is secretory unit is used to call this particular unit, it is where the secretion takes place. For example, if the secretions of these cells secreting within this luminal area, secretion is different. For example, this secretion is very corrosive when it moves from this particular duct at the time. So these cells will prevent the damage of the corrosive elements. So for example, the pH is maintained by this particular cells, and the secretion. Sometime what happens, the secretions required more, some in some gland, the secretion from this gland and secretion from this gland will satisfy the particular organ, right? So this is how the uh, gland secretory unit opens into the ducts, and that mainly opens into the main duct. So this is how the passage of the glandular epithelium will be there. So this is the best example. The figure is showing very clear cut. So this is the alveolar sacs, multiple alveolar sacs we have shown, and within this alveolar sac we have the glandular area. So that glandular area is branching, forming the branches, multiple branches, joining with all these branches joining with each other from the particular alveolar sacs. From there, forming the main tubule, it is we used to call intercalated tubule, and which ultimately drains into the salivary duct. So when you see the salivary duct, this particular salivary duct, so cut section at the time, so you may appear this particular figure. So where it's like pyramidal cells, lumen is larger, lumen is larger, and the nucleuses are centrally placed, right? This is very important. The salivary duct, the lumen is larger, pyramidal cells pyramidal cells and centrally placed nucleus, whereas the salivary portion, when you come to the salivary portion, the cells are uh, not numerous, they are scanty cells, and lumen is very narrow because it is somewhere else taking in the bifurcation part over here. So that area we shows there is a narrow lumen, it means very few cells are secreting and required very less luminal area. So when you see the some area of the face, exactly, we have the mucus gland, along with the secretory gland we have the mucus cells also, and these mucus cells showing that uh, the peripherally there is a nucleus, flattened nucleus, and it is basement situated on the basement membrane. So that is the main peculiarity about this particular cellular element, where the mucus, the jelly like substance which push the nucleus towards basement membrane. And important thing, whenever there is a release of this mucous membrane, so this apical part will get rupture and releases within the luminal area. That is the reason we have the nucleus towards the peripheral. Maybe we have, that is the main reason where the nucleus is shifted or scattered towards the basement membrane, right? Now you can see this, how the secretory vesicles which are present, exactly the pinkish discoloration. And you may appreciate there is a lumen, short lumen. It means we have 1 2 3 4 5. So there only 5 cells are there within that particular serous acini. This particular acinus is having peripheral nucleus. See, you can see the peripheral nucleus. Nucleus is situated in the peripheral area. It means it is a serous acinus which is situated peripherally, which constitute of or maybe the secretory part of your mucus secretion. Now, as I already told, there is an tubular glands, and I have told there is the best example is your gastric glands. So you see the crypts of lubricants, where the dot areas, so that is, those are the mucus glands, are empty cells we used to call. Empty cells is nothing but an the mucus or mucus gland. So when you see the most lower part will have the continuous. So these are the gastric glands, right? So this is completely in between the two glands. We have the lamina propria. So that lamina propria and along with this particular gland, they are situated over the muscularis mucosae, right? It means there is a mucosal layer. Within the mucosal layer, we have the glandular area. So that glandular area is used to call gastric gland, and they are situated over the muscularis mucosa, right? So this is how the example of the secretions of this particular cells releases within this particular tube, and these tube exactly releases within the intestinal flora. So that is our gastric flora. So that is the reason these particular glands which are situated is used to call gastric glands, and in the form of the crypt is, or the passage or duct is used to call crypts of lubricants, right? Now when you move to the classification of this particular joint, so as we already based on the site of secretion, for example, where that particular organ is secreting and where that particular organ is situated in our body. According to that, we have classified over here. That is, we know the exocrine gland, and another is endocrine, and the another one more is, that is, we used to call the paracrine gland. So these paracrine gland secretes produces into the local extracellular space affecting the surrounding. Example, anterior endocrine cells of the gastrointestinal tract. Just now we have seen. Now, what is endocrine and what is exocrine? Endocrine, already we know that the secretions from the organ, or else the glands which secretes from one place to moves another place without tubes. It means this particular secretion flow through the blood vessel, or else that is 100% the hormonal action. Whereas the exocrine gland, the secretes along with the tubes, or else we used to call ducts, from one end to another end, and these ducts are not so lengthy when compared to. So they are related to one organ to another organ, or one organ to, mainly in the GI tract we may find the maximum number of the exocrine glands. Now, how these particular exocrine glands, the secretions from these particular cells, the cells are here appearing columnar R, and from columnar to slowly they are decreasing into the cuboidal, right? So this particular secretion of the whatever chemical which produces by this particular gland, so this particular, earlier it is a saccule, from there it become a tubule, from there it opens into the outer surface, right? So when it comes to the outer surface, the whatever the other cell secretes, they'll get the chemical secretions, those secretion, and it gives the particular interaction. Now when you come to the classification of joint based on the number of cells, yes, definitely some gland have their own single cells. For example, we used to call unicellular gland. Example is goblet cells, which are present in the respiratory and intestinal tract, whereas multicellular glands, all the glands other than the goblet cells, they are multicellular. Multi means there are n number of cells will be there, they are forming a gland, whereas unicellular is a single gland which is present within the intestine or respiratory system. Why this goblet cell? Goblet cell secrete the mucus to prevent and protect that particular nearby epithelium, whatever they are secreting, or it, ultimately we may say it maintain the, it maintain the homeostasis, or else to prevent the damage of that particular epithelium. What it? Now classification of the glands based on the number of ducts and shape of the secretory end piece. For example, a simple gland may be having the one duct, as soon as it releases and definitely it opens into the effector organ. So only single. The compound gland means it may have the minor or major ducts. For example, when you see the your parotid gland, where are multiple small ducts which opens into the ductules and which ultimately form a organ. So so that is we used to call compound organ, compound ducts, glands. And this particular compound may divide it into again tubular or alveolar acinar and tubulo-alveolar acinar. It means they may have the tubulo-alveolar acini. It means the single gland along with the multiple uh, tubules which opens directly into the effector organ. Or sometimes what happens, the tubules along with the alveolo-acinus, the single united with each other for the multiple acinus will be there, which opens into the alveolar sac and which ultimately opens into the ductules. This is just, you can see the exocrine glands, simple types. So this is tubular gland, we may say. It means it looks like a tube only. The glands here, it is almost like a tube and which opens into the outer surface. And the same tubular some gland, but which is having branched, right, multiple branch. So this is tubular branched gland or branch tubular gland. And the what happens, this exocrine gland, rather than it is having tubular, but rather than multiple, it is a coiled single one, right, it is coiled tubular. And what happens, the instead of tubule, now the sacs are forming by means of alveolus. So that is the reason we have the alveolar branched, not single, alveolar branched gland. And you see there is a compound types, a tubular gland. Instead of one tubule, now we have the multiple tubules and multiple glands, but in the form of only the tubular area only, right. Now the multiple tubules, but along with the alveolar sac, right, multiple alveolar sacs. So this is how the exocrine gland, which is present in the multiple form of the various glands in our body. Now when we move to the multicellular glands, so this multicellular glands example just now I have shown, this is how the another photo I am showing you here. And this is how the compound tubulo-alveolar, the compound tubulo, compound means 1 2 3, there are multiple tubules and there is a multiple alveolar sacs which are connecting to the same tubule, right. Tubulo-alveolar, here it is tubular as well as alveolar sacs, tubulo-alveolar and compound tubulo-alveolar. So this is a best example how this particular compound tubulo-alveolar, alveolars and sacs. As the color of the gland is shown something and tubular epithelium is shown something different. It means the secretions also varies from those particular cells. Now the compound glands, it is a simple alveolar, so it is also called exactly seen in the penile urethra. So where single alveolar sacs will be there. That is a simple. One more, that is a simple branched alveoli. That is a sebaceous gland of the skin. Underneath the skin, wherever we have the sebaceous gland, they have their own branch, but it is a simple alveolar directly which opens. Whereas the compound alveolar, exactly the pancreas, parotid, mammary gland, glands of the respiratory system. So these all multiple glands which have the multiple alveolar sacs, and that opens ultimately into the tubular format. Whereas simple tubular, when you come, crypts of Liberkian, which we have studied in the stomach epithelium, right? Exactly, or gastric epithelium. So where we have the crypts of Liberkins. Whereas simple branch tubular, so that is an uterine glands. Remember, so this is an uterine gland or pyloric and fundus of the gastric glands, that may use to call simple branch tubular uterine glands, pyloric and fundic glands of the stomach, that is simple branch tubular gland. Whereas compound tubular, the Brenner glands of the and cardiac glands, where the Brenner glands are present in the duodenum, and cardiac glands exactly present in the lower part of the esophagus or else in the upper part of the stomach. So we may appreciate the cardiac glands. So they are compound, it means they are multiple in the form of tubule. Whereas simple coil tubular, exactly the sweat glands, they are just beneath the skin. The multiple coiling of that particular gland will takes place, and it is in the form of tube, which are present just beneath the epithelium of the skin. So there is a compound tubuloalveolar, submandibular, and sublingual salivary glands. So this is the best example anybody can compound tubuloalveolar. So this is the how the mixed salivary gland which appreciate just now. Classification based on the mode of secretion. For example, different cells secretes their different mode. For example, when you take the merocrine gland, merocrine gland example which is present in the pancreas. So such cells are uh, no loss of cytoplasm. There is cytoplasm. Example, the most of the compound glands. Example of pancreas, then there will be no loss of cytoplasm, but they secretes by means of diffusion. So this is used to called merocrine. Apocrine, what happens? Partial loss of cytoplasm. Example, lactating mammary gland. It means the lactation, whatever it is secreting within the that particular gland, which eject and comes outside, and which definitely opens into the tube. And the sweat glands, apocrine, the whole cytoplasm will come outside. The partial loss of cytoplasm. Axilla and external genitalia, wherever there is a sweat takes place, in that particular sweat, the secretions within the cytoplasm will come partially. Now, the what is holocrine glands? The complete loss of cytoplasm from that particular cell, the whole cytoplasm evacuate outside. For example, sebaceous gland, the sebum will come outside, or else the tarsal glands. So this is how complete evacuation from the your cellular element, that is the cytoplasm, evacuate from the that particular cell, that is we used to call holocrine. Now the cytochrine glands is nothing but an the cells are released a secretion. Example, testis, the spermatozoa. What happens? The cell content will remain same but produces a new gamete or new cell from there, that is extend outside from that particular cellular element. So this is we used to call cyto. Cyto is pertaining to nothing but an the new formation of a cell is used to call cytocrine cells. It means based on the secretion we have merocrine, there is a no loss of the cytoplasm. Where we have apocrine, there is a partially loss of cytoplasm. Holocrine means completely evacuation of the cytoplasm will be there. Now the cytokine within the cell, the one more new cell will get produced. So this is how this is used to call classification based on the glands. Now you can see the modes of secretions. Now you see exactly over here from the cytoplasm, the only the content release is the merocrine, right? Now the partial which is coming outside apocrine, right? Partially which is, and you see oh exactly the complete cell is getting cell cytoplasm is opening, so that is we used to call helocrine. So these are the best example how it is releasing from the cellular element, the cytoplasm release. Now when you come to the classification based on the nature of secretion, uh, as we know there are different serous, we know mucus, we know mixed. So these are the gland, how the what they secretes based on that. For example, when you see the serous gland, it is a thin watery secretion rich in enzyme. Example, parotid gland is best example, which is it means the thin watery secretions as good as the you may appreciate whenever we hungry and we see the good tastiest food, automatically the watery discharge releases in the mouth. That is exactly by the serous gland example, best example. The mucus gland, the secretion is thick and it is viscous like a blood, viscous secretion for the protection of lubrication of sublingual salivary. Example is sublingual. Now where exactly? So whatever the secretion from the gland which is uh, what we used to call the pH is more or low at the time, to prevent that we have the mucus secretions to prevent that particular, or else whenever we eat the very spicy food, to prevent that we releases the mucus, so that mucus protecting this particular part of the your epithelium, so that is we used to call. Whereas the mixed gland, so the best example is your submandibular salivary gland again. So where it is having both the watery and viscous, it means the different alveoli having their own secretions. For example, they have salivary glands even, and they have the mucus gland too. Now the difference between the serous and mucous acini, uh, it is there in your histology, any histology book. You may get this particular differences between the serous acini and mucous acini. Why should we know this? Because the mainly at the time of in the examination, when we wanted to find the serous secretions glands and mucous secretion gland and their staining exactly within the staining of that particular slide, how it looks. Now I have displayed this is the serous gland secretion and these are the mucous gland secretion. The one must one should there, the serous is having zymosan granules in cytoplasm, whereas the in the mucous gland the mucogen droplets, and the nucleus is centrally placed in the serous, and in the mucous gland it is peripherally. In the serous gland the lumen is small, whereas in the mucus it is larger lumen. The boundaries are not defined in the serous, whereas the boundaries are well defined in the mucous gland. The serous gland darkly stained with the your eosinic effect, whereas the lightly stained because of the rich in the acidic. That is the reason it is not stained properly. So the best example for the serous and mucus is, serous is parotid gland and mucus is sublingual gland is the best example. So this is how the mixed salivary glands. So this is where both serous and mucous glands are there. So which is having, this is the, when you see the serous gland, the lumen is more, and how the centrally placed nucleus, right? And you may see the semidominules which are present exactly over here. So this complete we used to call serous acini, right? And this is the mucous acini. When you see the lumen is smaller, nucleus is peripherally situated. So where exactly these two present? They present where there is an multiple organs having both the serous and mucus. And these serous, if at all you want to find, you may see the nucleus is centrally placed, and over that we have the semi-demi cells, acinar cells will be there. This is one more figure where we can appreciate the changes of the duct cellular changes, where the lumen is more larger and the nucleus is how it is situated, and centrally situated nucleus, peripheral nucleus situated in the right. When you come to the general architecture of the compound gland, so gland may be divided into, as we already in the lobe, when you come to the lobular area, lobes and lobules. So that lobes, when you see the parenchyma of that particular gland, is that may be the secretory end pieces, that is acini and tubular, or tubular or acini. And we have seen the ducts, that is the interlobular, intralobular and mare, that is the intralobular, interlobular and mare secretory duct. So that is about the parenchyma. So when you see the stroma of that particular cell, then we have the capsule which is encircled the complete the duct or else your alveolus. So that again guarded by a septa, that is you interlobular or intralobular, and we have the loose interlobular connective tissue supporting to the parenchyma. It means from the secretory part to the protecting part, both the parenchyma and stroma are connecting with each other to give a proper shape to this particular cellular element. So that is how the a compound gland should have the parenchymal and stromal cells arrangements. So the when you move to the clinically, adenoma, it's a benign tumor arising in the gland. Adenoma. Remember, this is very important. Adenoma. Oma is nothing but an tumor. Tumor. Adenoma. Adenocarcinoma. Right? Adenocarcinoma. A malignant. Oma is tumor and localized. Adenocarcinoma is a malignant and it is uh, connecting to the multiple areas. So either we used to call malignant is nothing but an where metastatic we is also called, right. So this is how the where mixed gland we are projecting over here. You may appreciate this picture is there in your book. You may appreciate and you can check the each and every content. There is an inter uh, septal area and within the lobular area, and how this particular adipose cells, empty cells we used to call, you can see the endocrine cells within that particular area. So here this figure is completely explaining all varieties of the glands, ducts and everything. So you can appreciate over this and you can compare the cell to cell, serous acini, mucus acini, everything is there in this particular. You may see over here exactly serous acini. Over that there is a semi-demian, right? And the stain is the bluish when compared to other. So the pinkish appearance, it is the mucus gland which is appearing like that. So this is intercalated discs, right? This is arterioles, and this is the duct, major duct, that is intertubular ducts. So see, this is the interalveolar ducts, right? So this is parotid gland, which is shown the how the serous glands and along with the serous. So this is the lumen of this particular and whole this substance is explained over here. This is a parotid gland. With the one more example, it is shown the striated duct along with the SNS. So this is the parotid duct. One more figure I have shown you. Interglandular adipose tissue in the parotid gland. Sometimes we have the in between the two cellular elements where with the uh, uh, adipose tissue that is also shown. So this is submandibular gland where the mucus acini is shown exactly, and in between we have the ducts, and this is a submandibular gland. So you can see the submandibular gland, the mucus acini is more, and this is the duct. How this ducts, you may appreciate the ducts over here. This is a mixed salivary gland where both we have the serous and mucus acini along with the ducts which within the interlobular area. Mucus acini. It completely appears like a empty cells only, right? But they are in nest-like appearance. So this is how the clearly shown the serous semidemules, mucus cells, and there is a striations of this particular capsular area and junctions between the right, exactly. This is sublingual, purely mucus gland, completely mucus. Septas you can appreciate over here, duct you can appreciate over here, and acini also you can appreciate over here. So this is one more sublingual gland. Minor salivary glands of the palate, exactly when you take the palate where you can aggregation of mucus acini and no striated ducts will be there because the direct mucous membrane releases within the oral cavity. How this the gland which appears exactly is the palate, the minor salivary glands aggregated in the encapsulated areas of the right. The one more figure I would like to show you. So this is exactly minor salivary glands aggregated unencapsulated areas, right? Exactly, these are the cells and empty cells, right? So this is highlighted in a high power which is shown. Dear students, so there is three major changes from the serous, mucus, and mixed gland. At all you want to appreciate, one should have to know the cellular elements from the alveolar to ductal or this system. If you read once the theory, then again we will demonstrate the slides in the histology. At the time, you will come to know that how these cellular element connecting from the central part of the organ to till to the peripheral where it pours the effector organ, that we may appreciate very properly during the histology classes. Definitely we demonstrate those particular slides, and we definitely may appreciate. And why should we know these secretions and everything? As already the clinically we may appreciate the normal size of the cells and abnormal cells to find out the adenoma and adenocarcinoma. So those are the things. Not only that, there are several small clinical conditions you may appreciate or you may analyze after knowing this particular pathology of this particular sites, right? Thank you, dear students. Thank you. You may, if you have any doubt, definitely can text the message or else you may ask the question. Okay. Thank you.
Thank you, sir.